So I'm Asfar Azmi. I'm Associate Professor of Oncology at Wayne State University. I'm the leader of Molecular Therapeutics Program at Karmanos Cancer Institute, which is an NCI-designated comprehensive cancer center. I'm also the Director of Pancreatic Cancer Research at this institution. And as a leader, my role is to look at how the bench-to-bedside science is happening at this institution. And I also help develop collaborative projects. So my area of expertise is drug development and translational research. And by translational research, I mean that the science that is done to move from lab into the clinical trials. In the lab, what we do is I lead a team of scientists who work on models of cancer, test new drugs, see their efficacy, and then move those drugs into clinical trials. And in the past, we have successfully moved many drugs from the lab into clinical trials. What I hope to accomplish with my research is to bring forward new and effective treatments for pancreatic cancer. Our goal is to enhance the survival of patients, and even if we could find a cure. And to do that, we are taking advantage of the latest cutting-edge technologies, incorporating them in our science, which allows us to transition the findings in the lab into patients for effective treatments. So the new technologies which we are using include better animal models that represent the disease, patient-derived models where we can grow patient tumors outside in the lab, and then a vast array of computational technologies that includes spatial biology, artificial intelligence, which are being used to dive really deep into the pancreatic tumor's biology and understand why it is so resistant, why it does not respond to drugs. We are able to design better drugs, design better combinations, which are tailored to individual patient tumors. They are tailored towards a certain mutation in those tumors, and that's how we feel that these tailored trials, which would be focused on a certain mutation, could be more effective than what was previously done when patients were getting chemotherapies or drugs. When it comes to how my research impacts the disease, we have in the last 10 years been working on a drug that is called selinexor. This drug targets the protein transport system in the body. Now, proteins require to be in the correct place to be functional and perform their tumor surveillance or anti-tumor activity. So we have developed a drug which blocks this protein transport, keeps the good proteins in the correct compartment. So when this drug keeps the proteins in the right compartment, then we see cancer cell death. We see animal tumor cell death, and we have seen different types of other models showing response to the drug. Now, when we did sufficient amount of preclinical work in the lab, we were able to move this drug into clinical trials for patients with pancreatic cancer. In those trials, we were able to see really good responses in some patients. We increased the overall survival of those patients. We even had improved quality of life of these patients on this drug. Right now, we are trying to understand why some patients responded, why some did not. This would allow us to find the appropriate patient population which would be most responsive to the drugs. And down the line, when we do future trials, we would only give this drug to the responders which we have identified in our lab experiments. What got me into the field of pancreatic cancer? It's a very good question. So personally, I like challenges. And we all know that pancreatic cancer is the toughest disease to target or cure. So I took up this challenge that I need to find something, in this disease. That's one part. The other thing is that I have some connection with pancreatic cancer because I have lost a friend to pancreatic cancer. And that is also one of the driving forces in my choice of disease as a researcher. I thought that this is an untested territory. This is neglected disease. And working on this topic would be more beneficial, and I could make a bigger impact as a scientist, while being lost in a big group of researchers who are working on those well-studied diseases. I may not be able to make an impact. This is another reason why I selected pancreatic cancer. How my research would benefit pancreatic cancer patients, it lies in the work that we do. We are a drug discovery lab. We are a lab which does translational science. We move concepts from lab to clinic. We work on several new drugs, new drug combinations. The whole idea about the work in the lab is to bring new clinical trials, which is acutely needed for pancreatic cancer. So right now, we have some very novel drugs we are working on beyond selinexor that I talked about. And we have been generating data which is convincing enough to the clinicians that they want to initiate trials on these drugs. So we hope to see more and more trials emerging from the work done in our lab. And it will not be just in pancreatic cancer, but it could be beyond this disease in other diseases as well. In terms of next steps, we are working on the KRAS pathway. KRAS is one of the most frequently altered pathway in most cancers, but it is more frequently altered in pancreatic cancer. About 80 to 90% of patients with pancreatic cancer have KRAS mutations. And what this protein does is it keeps the tumors in a constant growth phase. Now, we know all this for the last 40 years, but there was a challenge in developing drugs against KRAS. Now, recently, with the advancement in sciences and technologies, drugs have emerged. So my lab is focusing on these KRAS inhibitors. We are trying to find how to combine this drug with chemotherapy or with other targeted therapies. We have also been one of the first labs in the nation to develop resistance models of KRAS inhibitors. So we want to preemptively understand that when patients get these drugs, how would they develop resistance and what would be the ways to overcome resistance. So down the line, when we will initiate clinical trials, we would be able to find the right patient population on one side and also the right combination to give to the patients for a very prolonged effect of the drug and prolonged survival of the patients. That's a tough question to answer, that if I have unlimited funds, what would I do with it? Well, I would continue my drug discovery efforts. I would find new and more effective drugs and test them and bring them forward to the clinic. But at the same time, I feel like it's my duty to also train the next generation of scientists in pancreatic cancer. So I would provide the top resources to junior scientists, students who want to work in this field, because we acutely need more scientists to work on pancreatic cancer. So if I get unlimited funding, I will use that wisely to train the next generation scientists and continue what I'm doing, and that is to find more and more therapies for pancreatic cancer patients. When I was a young investigator, I was struggling to develop my own grant portfolio. I did not have the funding to do research, and usually the federal agencies want the investigators to submit a very advanced proposal with a lot of data in it. I did not have the money to do that. So I was struggling, and in comes Sheila Kesselman from Sky Foundation. She had a habit of scouting for the investigators who were trying to establish themselves in pancreatic cancer. So she came, she talked to me about what my project was, what its potential was. I explained everything, and then I received funding from Sky Foundation. So this is how my interaction started, and this started more than 10 years ago. Yeah, so my story with Sky Foundation has been a very successful one, and I was working on this topic of nuclear transport, which I touched upon in the beginning. We were developing this drug, and we were trying to apply for federal funding. But each time we submitted a proposal, we used to get back these comments that we need more data to convince us. So when Sky Foundation came to me, and we had a nice interaction about developing this project further using the seed funds from Sky Foundation. And the funding that I received, I used it to develop more data. We revised the proposal, we submitted to NIH, and then we were able to receive 1.5 million funding for one project. Then separately, I was working on another project, and Sheila Kesselman also funded that, and I got a second grant. So with the seed funding from Sky, I was able to get more than 3 million of federal funding, which is, I would say, a success story. Sky Foundation has really impacted my research. In fact, I would rephrase it and say that I owe my research career to Sky Foundation for the seed funding and the funding at the critical time that they provided me, and that helped me grow as a scientist. And I think that for me, Sky Foundation means hope. The amount of work they are doing for the community, spreading awareness about the disease, funding investigators like me, has a very long-term and impactful. People never used to talk about it 10 years ago, but with this foundation, we now know about this disease. People talk about it. People think about going to the doctors earlier when they have a symptom. And Sky Foundation has done an excellent job in that. On the other side, as researchers, we benefited immensely, and I'm now seeing that not just locally, but Sky Foundation is doing work nationally. When I meet the Sky Foundation people, I see a genuineness in the whole group, and I see the genuine efforts, which is very moving and touching. And when I talk about these activities to my kids, they also say that we want to be part of something like this and grow up and do something for the patients. So this is my personal view, that I feel that I'm blessed to be in contact with Sky Foundation, and it turned out to be a very successful story.